So, getting started, I'd like to introduce today's speaker, Jeremy Stovall. Jeremy received his bachelor's in Environmental and Natural Resources from Clemson University, a master's in Natural Resources Forestry from the University of Vermont, and a PhD in Forestry and Forest Products from the Virginia Polytechnic Institute and State University. He's currently an assistant professor of silviculture at Stephen F. Austin State University. Jeremy?
All right, thank you, Sean. So as we go through the talk here today, please feel free to interrupt me with any questions you might have. If I'm not watching the discussion board, Sean's going to keep an eye on that and will interrupt me so we can address those as we go. Today we'll be talking about nutrient cycling and nutrient deficiencies in production forest, primarily focused on loblolly pine forests in the southern U.S. We have three key objectives we're going to try and meet today. First, we're going to look at what limits growth in southern pine forests. Second, we're going to look at how nutrients play a role in that and how they cycle in these forests. And finally, we're going to look a little bit about how climate change and some of the effects it may have on our forests will actually alter forest nutrition and how that may impact growth in the future. We're going to be looking specifically at how changes in atmospheric carbon dioxide concentrations may impact forest nutrient cycling. Okay, and throughout here I've got a couple interactive quizzes just to get everyone's input on where we're sort of starting at and what your thoughts might be. So Sean may have directions for us on how you can go about and actually complete this, but right now let's look at this question. What do you think most limits growth in pine plantations in the southern U.S.? Is it A, water, B, nutrients, C, temperature, or D, soil physical properties?
And to answer that question, right under your name, under the participant sidebar, you should see 4 icons, and the one with the little A will let you select what answer you'd like to submit. So we'll just give everyone a few seconds to submit their answers.
We got about half completing this, but we've got a number of moderators in here as well. So far it looks like about 2 answers for water, 2 for nutrients, nobody thinks temperature, and then 4 for soil physical properties. So we're starting off with some combination of water, nutrients, and soil physical properties. In part, you know, obviously this is a large area, the southern U.S., and there's going to be some variability, but let's look at some data that can hopefully start getting us closer to figuring out what the actual limitations may be. So what I want to show you here are first a couple photos, and then we'll look at some data. This is an experiment called C-TREES, which is an acronym for the Southeast Tree Research and Education Site. And this is located in the Sandhill regions of North Carolina. It's a collaboration of the Forest Productivity Co-op, the USDA Forest Service, and a number of other partners, and has been running for several decades now. And so what they did is they took an industrial forest plantation, and I believe it was at age 7, they installed 4 different types of treatments in a replicated experimental design. And so what they did is they left some areas untreated. Some of the plots they fertilized only, some of the plots they irrigated only, and then some of the plots they added fertilizer and irrigation. When you look at this soil, what they did is they picked a site that they thought was absolutely as nutrient and water deficient as they could find. This is a very deep, very sandy soil. It does have some clay lenses at 2 to 3 meters depth, but they were really trying to find a site where either water or nutrients or both would be limiting as much as possible. These photos that I'm showing you right here on the left I took at the Silviculture Instructors Tour last October, and so this is 20-some years later. And what you see on the left is a control plot, and what you'll notice in the foreground right here, that I'm highlighting now hopefully with the laser pointer, is actually a longleaf pine. And so in the control plot where they added no water or nutrients, you can see there's a lot of open sky there. There's been a lot of density-dependent mortality. A lot of trees have died. They're not competing very well, and longleaf has actually remained competitive with the loblolly pine that was planted. On the right, this photo I'm showing you here is a fertilizer plus irrigation plot looking straight up. There are no longleaf pines in that site. The diameters on the tree are much larger. The heights are much, taller. There's much more volume production, and you can see it's a lot of leaf area there. The trees are supporting a lot of leaves. And so if I flip to the next slide here so we can look at this actual data, this data was published in 2004, so it takes it out through age 16. These trends have continued as you see them. But what we have is this blue line right down here. That's the control plots. This red line right up here, that's the irrigation-only plots. This green line right here, that's the fertilizer-only plots. And then this purple line up here is where they fertilized and irrigated. Lee Allen and the other researchers that were conducting this project thought for sure that they were going to find that this site was going to be water limited. But what this data is telling us is that on this very sandy, very nutrient and water deficient soil, nutrients are actually more limiting than water. We can't get a handle on which nutrient specifically with just this data, and their fertilizer treatment was actually a blend of macro and micronutrients. So they were trying to handle total nutrition to ensure that no one nutrient became limiting. But in looking at this, they initially came up with the theory that nutrients, in fact, are probably more limiting in the southern United States than water for growth. If you start thinking about it, that does make sense. On average, anywhere from East Texas here in the West Gulf, where I'm at, all the way over to the Carolina coast in Virginia, you're getting in excess of 40 inches of rain a year, more than 44 inches of rain a year, all the way up to 50-something inches of rain a year. And so this is an area that gets a lot of rainfall. And so if you think about it in that context, it may make sense that nutrients are more limiting. I should point out that if you're in the West Gulf region here, so East Texas, Arkansas, certainly southeastern Oklahoma, our evapotranspiration rates are significantly higher than this study site. They actually looked at some of this early data and found that they were only developing a water limitation for about 9 days a year at most, and those days weren't contiguous. So we may get into periods here in the West Gulf where we are actually water limited for a time, but over most of the range of loblolly pine, and certainly where it's planted as plantations, it seems to be that nutrients are in fact more limiting than water to growth, and certainly than soil physical properties. As we talk about nutrient limitations in production forests and water limitations, you have to keep in mind Liebig's law of the minimum. And a simple way of stating that is what limits growth the most is going to be the most limiting nutrient. And one good way to think about that is the analogy I've got here on this photo, which is a barrel. In this context, you can see a different nutrient or requirement of a plant like water is written on each stave of this barrel, and the staves have different heights. In this analogy, the staves represent a nutrient or water limitation, and the water in the barrel itself represents overall growth. And so as you can see on this photo right here, the lowest stave happens to be water. This would be an example of a water-limited site. And so you could add all the nitrogen, for example, right here that you wanted. You're not going to get a large growth response because you haven't fixed the problem with that stand, which is a water limitation. Conversely, let's pretend that this water stave was very high and we did not have a water-limited site. Nitrogen, you can see, would then be the next most limiting nutrient. If you applied 200 pounds per acre of nitrogen fertilizer on this site, you might see a small growth response because you basically fixed part of the deficiency, but very quickly potassium becomes deficient. And so unless you fix all of the various nutrient deficiencies that a stand may have, you may not see a substantial growth response. So it's important to keep in mind when we're looking at data what was actually limiting nutrients in that stand, what data we have that supports what was limiting nutrients in that stand, and whether those are limitations that were fixed or not. So you can't just look at purely nitrogen data and necessarily be able to tell that that is what was limiting growth or not. So if I flip back real quick to the sea trees data, you can see they accounted for that by having an irrigated plot, a fertilized plot, and then combining them. So they were accounting for Liebig's law of the minimum. When we apply some of these concepts we're starting to talk about here to a pine plantation, we also have to think about how the stand develops over time. And we have a very simple and convenient model here to think about how nitrogen deficiency may develop in a forest over time as that plantation is established in an old clear cut. And so we have about a 24-year time scale here, and what you're seeing is an initial high flush of nitrogen. See this large red peak in the soil supply? Well, that's going to be your slash decomposing from the previous clear cut, which happened at time 0 in this example. So early in a plantation forest, you have a lot of nutrients, a very high soil supply, and you have low stand potential demand. We've all seen young plantation forests. That makes a lot of sense. The trees are occupying very little of the site because they're small, they haven't closed crowns, and their nutrient requirements are still very minimal. They're primarily rooted in the A horizon if there is one, and so there's lots of available nutrition in basically the entire rooting zone. As the stand continues to grow, the slash decomposes, releases its nitrogen that's uptaken by either your crop species or often competing species on the site. On very sandy soils, you may actually have some nitrogen leaching through the soil profile, but by and large we don't see a lot of that in forests since they're deep-rooted systems. And at some point, your trees are larger, crown closure begins to occur, and the trees need more nitrogen to continue growing, and right around here in this example is where our nutrient deficiency would develop in age 5 in this stand, which would be a fast-growing, slightly probably above average stand. We see another pool of nitrogen emerging for the plant here, this remobilized nitrogen. That remobilized nitrogen is largely accounting for the forest floor. So after a clear cut, what pine needles are out on site decompose. They release their nitrogen over about the first several years, and they're not there anymore. Following crown closure, you start building that litter layer back up over time, and that, if you want to think about it this way, that the tree fixes that nitrogen, it puts it into the needles, it uses them for photosynthesis. For a southern pine, you're looking at somewhere around 2 years it hangs on to those needles for, and then it retranslocates that nitrogen. It pulls as much of it back out from the needles as it can, and then the needles fall, but it can't get them all. And so as that forest floor builds up and continues to decompose, and there's a lot of fine roots in the upper A horizon and even in the litter layer, the trees recapture that nitrogen that they'd already uptaken from the soil. And so we end up with a slightly higher availability of nitrogen than just what's uptaken from the soil. But look at this blue dotted line here. This is your stand potential demand, okay? And that difference between this dotted blue line and the top of these red and green bars here, the actual available nitrogen, that's basically an opportunity. That's nitrogen that could be growing your stand at a much greater rate if it was available, and that's where fertilization comes in. You can add fertilizer on top of this remobilized bar and actually get greater growth rate from your pine plantation with a silvicultural treatment. And so this is why fertilizer works in southern pine stands. We have nutrient limitations, and it can ameliorate those. Now when we're talking about nutrition in trees, what we've figured out works pretty well as a way to understand nutrient limitations is a concept called leaf area index. The saying that Lee Allen came up with at the then Forest Nutrition Co-op, now Forest Productivity Co-op, was nutrients grow leaves and leaves grow trees. And so looking at the amount of leaves that a forest stand has is a good surrogate for estimating perhaps whether it's nutrient limited or not nutrient limited. And so when you discuss leaf area index, I've got a very simplistic diagram here. All you're thinking about is the area of leaf above a given area of ground. And so whether that's in units of square feet of leaf per square feet of ground or square meters of leaf per square meters of ground, it ends up being a unitless ratio. And so here you see an example of a leaf area index of 3. On the right, you see an example of a leaf area index of 1. This is, of course, more complicated to actually estimate in a growing forest. Pine needles, if you look at them, a fascicle of pine needles is actually round in cross-section. See if I can draw it out here. I'm working on a touchpad, so this may not be very geometrically precise. But if you look at a cross-section of a fascicle of pine needles, it looks like that. If it's loblolly with 3 needles per fascicle, you've got 1 needle there. So there's needle 1, needle 2, needle 3. And so when you actually go to estimate this, you need some technique to basically estimate the total surface area of that entire pine needle, scale that up to the whole canopy, and figure out if you're going to look at a one-sided leaf area or a two-sided leaf area. As you can imagine, this is going to be simpler in hardwoods, broadleaves, since their leaves are much more similar to what I've drawn here. We won't go into too much detail. I'll give you a little more detail later on how we can do that efficiently over large areas. Suffice it to say there's a number of different techniques, and they all get some rough estimate of leaf area index that can compare stands that have low leaf area indices to stands that have high leaf area indices. And this is the reason why we're doing this on this figure taken from one of Tom Fox's paper. What we generally find that with higher leaf area indices, as you would expect, those trees and that stand is able to capture more light. As a tree captures more light, it's able to grow at a faster rate because it has more sugar that it's obtained through photosynthesis that it can then allocate to biomass production and respiration. And so we see a reasonable range of leaf area indices shown here from 0 on over to 5. And here's a very typical average in the middle here at 3. And what you'll notice at a leaf area index of 3, we get about an average of 300 cubic feet per acre per year in terms of our current annual increment. Now you'll notice these two dotted lines here and here bounding the mean line right there at 300. What those lines are representing is variability. And a lot of that variability, we believe now, is attributable to differences in growth efficiency. That growth efficiency can come about for a number of different reasons. Some of them may be stand factors. A stand growing at a very high density, say 2,000 to 3,000 trees per acre, may have relatively low growth efficiency because those trees are all extremely stressed compared to a stand growing at a more normal plantation density of about 500 trees per acre. So you have stand level factors. Another factor you may want to consider would be a genetic factor. So if you look, slash pine and loblolly pine have different growth efficiencies. So we can consider genetics from the standpoint of a species with a different growth efficiency. The other thing we can consider is differences in growth efficiency within a species. You may have differences between unimproved seeds and seed sources improved through genetic improvement. They may have seen increases in growth efficiency. When we move to varietal or clonal forestry, we're really observing large differences in growth efficiency between individual clones of loblolly pine. Some of these varietal trees can grow at incredibly fast rates, even though they maintain a relatively small crown. And so these are just genetic individuals that have very high growth efficiency due to efficient use of nutrients, greater efficiency of photosynthesis, or some other physiological attribute. And so genetics is one major factor that determines this variability around the mean. But in general, you see the overall trend. More leaves are going to grow you more wood. And what we're trying to primarily manipulate with fertilizer additions is adding more leaves to those trees so that we can then produce more timber volume. And if you look at what's been going on in the U.S. South, this data runs from 1990 to 2004, you see that industry primarily has realized that nutrients do in fact grow trees, and they've responded by fertilizing more than half of our currently about 34 million acres of loblolly and slash pine plantations in the U.S. South. So you're talking about somewhere in the vicinity of 16 to 20 million acres that has been fertilized at least once since it's been a forest, primarily with nitrogen and phosphorus. You see a lot of that is nitrogen and phosphorus at establishment, more and more based on our understanding of this dynamic I showed you right here, where nitrogen may not be deficient early on. We're seeing more and more of a shift to later fertilization rather than necessarily just fertilization at establishment. The other thing you'll observe here, it rapidly picks up in peaks about 1999 and then starts dropping off. A lot of that drop-off is simply attributable to increases in fertilizer prices. Nitrogen fertilizer costs are very closely linked to the cost of oil. And so as oil goes up, fertilizer becomes more expensive, and industry focuses more and more on fertilizing less if they can get some growth response out. It really comes down to the financial analyses more than just what the biological growth response is going to be. On average, across the South, with all this fertilization, we see a 25% growth response to a mid-rotation application of nitrogen fertilizer, with a typical response duration of somewhere around 6 to 10 years. So our stands are on average responding well. That's the average. There's wide variability around the mean, ranging from 0% to 100% or more. And so there's an enormous amount of variability in terms of how specific stands will respond to fertilizer addition. Okay, so that's wrapped up objective 1. What are the limitations to growth in our southern pine forests? And I would argue from the literature, by and large, it's primarily nitrogen and phosphorus, followed by potassium, micronutrients such as boron, and water. Now, of course, I've got the asterisks there. We're speaking in broad generalizations here. You may well have a site here in East Texas that is primarily water limited. You may have a site on an old Pleistocene terrace on the lower coastal plain that is primarily phosphorus limited. There are certainly examples of soils in Florida flatwoods that are primarily boron limited. And fixing any one of those limitations could result in massive increases in growth. So we have a wide range of sites, but on average, generalizing across the South, nitrogen and phosphorus appear to be our greatest limitations to growth in our production forests. Does anyone have any questions on this first portion of the webinar? Again, if you came in late, please feel free to interrupt with questions at any point in time. But if I don't spot those, Sean will point them out to me, and we'll stop at any point and go over any questions you may have. If there's no questions, let's move on to our second objective, and we'll start that with another of these pop quizzes. So I've got 2 here. This first one is going to be, which nutrient is the least mobile, the least able to move in the soil, and thus probably the most difficult for trees to obtain? And I've got 5 choices here for you: A, nitrogen; B, phosphorus; C, potassium; D, calcium; and E, magnesium. And I think a few people may have joined us late, so Sean, maybe you want to go over the procedure for making a choice here quickly again?
Absolutely. Right under your name, under the participants bar on the side, left side there, you should see a little icon with an A in a box. If you hover over that, you should be able to select whatever answer you'd like to submit. And we'll give everybody a few seconds to go ahead and answer.
Well, folks are thinking about that. It looks like we've got a question from David Fox, who asks, going back to your original poll, is it not soil physical characteristics that regulate nutrient and water availability? Soil texture absolutely plays a huge role in nutrient and water availability. So yes, you could certainly make that argument, particularly if you're focused on texture. If we look at other soil physical characteristics, such as soil strength, for example, what that really impacts is the ability of trees to root, which then, of course, is going to impact their ability to uptake nutrients and waters, pan layers, things of that sort. And so I would argue that nutrients are probably the ultimate cause of slow growth in that case, nutrients or water, but soil physical characteristics can certainly cause those deficiencies to develop if you're developing a poor rooting habit. When we look at soil physical characteristics too, we have to consider how tree roots work, and they're really quite remarkable. Tree roots really are good at finding the path of least resistance. They're basically growing under turgor pressure and nothing else, which is just water pressure. And so it's very analogous to attempting to blow up a water balloon into the soil. That's how they're growing. And so they've developed a number of different traits that allows them to find the path of least resistance. So if you clear cut a pine plantation and put another pine stand in there and you're planting your trees, you know, halfway between your old rows, and you go back and dig some of those trees up, what you'll find has happened is they've rooted over into those old stumps as they decompose. And they'll actually root down into the old stumps because there's low soil strength there and lots of nutrient water availability. And so it behooves them to grow there. The other thing you'll see is in soils where you have differences in strength, where there may be a path of least resistance between, you know, otherwise strong soil, something like where you've ripped to break up an old plow pan, you tend to see the roots following that. And so it's easy to get into a trap of thinking about roots just uniformly growing everywhere throughout the bulk soil, but really they're going to grow preferentially to areas with lower strength. And so for that reason, you know, even in a soil with poor physical characteristics, not speaking of texture but strength, it may not limit growth as much as we might think it would. Of course, again, this is all generalizing everything a lot, and so you could find a scenario, especially here in Louisiana or Texas or parts of Arkansas or Alabama, where you have a lot of vertic soils or vertisols with shrink swell properties. That's obviously going to be very challenging for a tree to grow in. So there are some examples where soil physical characteristics would absolutely limit growth. Did that address your question, or do you have a follow-up question, David? Okay, feel free to type it in if you have one. Let's look at this poll here, and it looks like we've got about 7 people for phosphorus, about 4 for potassium, and then a number of us who haven't responded. And so we'll look at the data on this, and I would argue, as many of you selected here, that it would be phosphorus, and we'll show you that data in a moment. I believe here, yes, I have a second question. Let's take a stab at answering that. And so what nutrient makes up the greatest percentage of a tree's mass? And you have the same 5 choices there. So take just a minute and let's see what everyone thinks about what nutrient makes up the greatest percentage of a tree's mass. Okay, it looks like we have 9 for nitrogen and 1 for phosphorus. So either everyone's already on the same page here, or perhaps you already downloaded the slides and have seen the next slide. In either case, let's move on and look. This is, again, overly generalized, but we have an idea of the chemical elements that make up a tree here shown on this table. When you look at this, you'll see that the top 3 choices that make up about 96% of a tree mass I did not include on that pool because we really typically don't consider them nutrients. So carbon on average is about 45% of a plant's dry mass, oxygen is about 45%, and hydrogen is about 6%. Those are all derived from either the atmosphere or water. If you think about where the carbon is coming from, it's really pretty remarkable. Carbon dioxide right now is 400 parts per million in the atmosphere, which is something like 0.04% of the atmosphere. It's literally a trace gas. But if you think about the largest redwood on the planet that's 378 feet tall, it's gotten that large basically by filtering a trace gas from the atmosphere through the process of photosynthesis. So really remarkable how forests grow. If you think about why those make up the majority of a tree, it makes a lot of sense. So any carbohydrate is basically CH2O in that ratio, and so that's what's going to go on to make up cellulose, lignin, and all these other biological molecules, organic molecules that make up wood. And so that's clearly the vast majority of a tree's mass. Even the lipids in a tree that make up the cell membranes, lipids are primarily long-chain hydrocarbons, and so they're made up of C, O, and H. The only reason hydrogen, there's more probably atoms of hydrogen in a tree than any other element. The reason it's such a small percentage of the dry mass is that hydrogen atom, remember back to basic chemistry, is just a proton and a neutron and an electron. Whereas carbon is going to be 12 of each, and oxygen is going to be 16 of each. And so you have basically just lighter molecules there. If we look at the elements, nitrogen makes up about 1.5% in terms of the first nutrient there, potassium about 1%, calcium 0.5, magnesium 0.3, phosphorus 0.2. There's some rounding error there. It's typically about just 10% of what nitrogen is, and sulfur at 0.1. You'll see this doesn't add up to 100%. I obviously haven't included every element here. But those nutrients, and we're considering them nutrients because primarily they derive from the soil. And this is why most of this talk focused on forest nutrition is really focused on the soil. So that's what a tree is made up of. And here's a little more detail on our key 3 macronutrients, nitrogen, phosphorus, and potassium, which are going to be the most limiting in a forest. Nitrogen being N, phosphorus being P, potassium being K. And so you see they work very differently in a soil. And understanding how these elements cycle in a soil is critical to understanding how they may be limiting the growth of our forests. And so if you look at nitrogen, it's really biologically mediated. It's available in a number of different forms, ammonia or nitrate primarily. In our southern forest, it's going to be primarily nitrate, which is an anion. And if you look at nitrogen, you know, the largest pool on the Earth of nitrogen is the atmosphere, which is 78% nitrogen. But remember, that's in the chemical form of N2, which is basically going to be nitrogen. Here, let me see if I can find the pencil here. There we go. Nitrogen triple bonded to another nitrogen atom. And so that's a very difficult bond for plants to break. We'll look at the nitrogen cycle in a minute, but they really don't have access to that atmospheric nitrogen. And so really, their only source of nitrogen is nitrogen that's already been biologically fixed and is present in organic matter. You have to keep in mind they're competing for that. They're competing with microbes, they're competing with fungi, they're competing with other plants, whether that be the pine next to them or, you know, the sweet gum or other competing vegetation you may have. And so biological processes are dictating how nitrogen cycles. And so really what we're doing when we manage nitrogen primarily is we're managing organic matter. This is why how you manage slash following a clear cut can have a big impact on how your next stand grows, because that's basically how you're managing the nitrogen. We can also manage nitrogen by applying either organic, such as poultry litter, or inorganic, such as some of our chemical fertilizers like DAP, fertilizers to a stand. On average, what we see at mid-rotation is that nitrogen fertilizer responses are short-lived, about 6 to 10 years, maybe a mean of about 8. Phosphorus works a little differently in the soil. Its primary form you see right here, uptaken by plants, and again, this is generalizing again, but phosphorus is cycled both biologically and chemically. You can have calcium, iron, or aluminum phosphate minerals, depending on what your parent material is, and so you can have some chemical weathering that releases phosphorus in some scenarios. In other scenarios, and more typically probably, phosphorus cycles like nitrogen cycles in that it's regulated by biological processes and is dependent on organic matter for phosphorus release, as well as, in the case of phosphorus, P that is absorbed or adsorbed to clay surfaces, and whether that's becoming plant available or not. Finally, here you have potassium, and that really is primarily derived from chemical weathering of primary and secondary minerals that have potassium in them, and it really cycles as a cation. So it cycles in a very simple chemical form, just as an ion. And so if you look at what these are doing in the tree, nitrogen is going to be critical in any protein, which is built of amino acids, all of which involve nitrogen. Without proteins, we don't have enzymes, and without enzymes, we don't have life as we know it. Chemical reactions in living organisms would be too slow. Phosphorus plays a diversity of roles, but one of the most important of which is helping to transport different molecules back and forth across lipid layers that make up the cell membrane. And so without phosphorus in that role, photosynthesis wouldn't work as it does, respiration wouldn't work, and you wouldn't have live trees. Potassium works in a plant very much like it works in an athlete when they drink Gatorade. So you always see Gatorade advertising electrolytes. Well, potassium is really a free electrolyte in plants. One example of where it's important in plants is in whether the guard cells around the stomata, the little holes in the leaf, that let carbon dioxide in so it can be converted to sugar through photosynthesis. Potassium plays a major role in how those guard cells work by serving as an electrolyte. And so we can control nitrogen with fertilizer application or by manipulating organic matter. We control phosphorus by managing soil pH. I'll show you a slide on that more, and I have not done in forestry there. It's rarely done in forestry. You'd have to often be on a situation where you have a short rotation woody crop, perhaps something like eucalyptus, before you really see much liming done to manipulate soil pH in forestry. Our rotations are too long really to make that economically viable. We can also manipulate it through fertilizer addition by chemical fertilizers such as triple superphosphate or DAP or other chemicals like that, or again with organic fertilizers such as poultry litter. On a phosphorus deficient site, a 50 pound per acre application of phosphorus in a chemical form can actually, we found, have a positive growth response to that stand for as much as 2 rotations. So a much longer potential increase in growth rates with phosphorus fertilizer application, and we'll see why as we look more into how phosphorus cycles in a forest soil. Potassium is primarily controlled through fertilizer application, since there's really very little we can do to alter the rates of chemical weathering of primary or secondary minerals. Okay. When we think about how nutrients cycle in a forest, we really need to think about what we call the QCI framework, and those stand for quantity, capacity, and intensity. The total amount of nutrients in a forest soil are not what a plant actually has access to. What a plant has access to, those plant available nutrients, are what we really care about, because those are what manipulate growth, what alter growth. The total amount in the soil isn't what necessarily alters growth. And so if we look at this, we're going to define quantity as the total amount of nutrient in a soil present. Capacity is the rate of supply of that nutrient to a plant. And so we just talked about what processes mediate nutrient availability for 3 of the major macronutrients. The intensity is going to be the actual amount of nutrient available to the plant at any given point in time. And so again, keep in mind, you can have literally a ton of nitrogen per acre on a site, but that doesn't mean that it's all plant available. The vast majority of that may be in a form that is unavailable to plants. So if we look at a typical example for nitrogen, these are some very realistic numbers for a forest. You may have 2,000 kilograms of nitrogen available per hectare. That's a very large quantity or pool of nitrogen. But if you look, of that 2,000 kilograms, only 30 kilograms per hectare per year are actually being made available as the capacity in plant available form. And so that's a much, much smaller rate you see now. What's going on there is basically decomposition processes, mineralizing or releasing nitrogen. And then when we look at the intensity, what a plant actually sees may be 6 parts per million of nitrate dissolved in the soil solution. That's what a plant can actually uptake through its roots. And so you can see, if you get a soil test and it tells you you've got 2,000 kilograms of nitrogen per hectare in your soil, that may have very little to do with plant growth on that site. You may still have a very nitrogen-limited site because you're not accounting for the capacity and you're not accounting for the intensity, which are what are actually more closely correlated to tree growth. And again, if you look at nutrient cycling, it's going to be complex. Here's a relatively simplistic diagram of how nitrogen cycles in plants and in a soil. And so we're not going to go through this in great detail, but I just wanted to show you this briefly as a refresher back to the forest soils, and just to point out that there's really a lot going on. There's a lot of soil chemistry going on. There's a lot of biological processes going on that are dictating how any nutrient cycles in a soil. And so with nitrogen, we've got a number of forms that are going to be less available to plants in the south, like ammonia, which is less plant available, nitrite, which is less plant available, and that all has to be converted to nitrate before it's really very plant available in general in our forest soils in the south. We have this large pool of atmospheric nitrogen that's not plant available, and we have certain biological processes, such as legumes, for example, that can fix nitrogen through a symbiosis in their roots, or nitrogen-fixing soil bacteria. Decomposers play a critical role in breaking down organic matter and releasing that nitrogen back to mineral form so that a plant can uptake it. So just a very quick refresher on nitrogen cycling. If we look at some typical soil orders, this may be the percent total nitrogen available in the soils versus depth. And so here you see a mollisol, for example. As you go from 0 to 30 centimeters, which would be a foot, 2 feet, 3 feet, you can see it drops from about 35% total nitrogen down to about 10%. Now, your mollisols are really your very nutrient-rich prairie soils, by and large. If we look at these bottom two down here, the spotosols and the ultisols, these are going to be much more typical of our forest soils in the south. You have a lot of spotosols in South Georgia, North Florida. Many forests on the southern coastal plain and Piedmont are going to be ultisols, many forest soils. If you look at these, you can already see a much lower nutrient availability. So in ultisols, 15%, on down to maybe 5% at 3 feet depth. Spotosols, 15%. You see this brief uptick right here about foot That's because in spodosols, if you're familiar with them, the organic matter moves downward into that spodictic horizon where you have greater organic matter and therefore greater nitrogen. And so it's all accumulated there, and then it drops off sharply down to only a few percent. Spodosols, in general, tend to be sandy in texture, so you have less available nutrients there. If we look at these diagrams, you can already see an advantage that trees have over annual crops like corn. There's a lot of nitrogen at depth that our trees will have access to that annual crops don't. The other thing you do see, though, is that the majority of nutrients are available in the surface soil. And so that's why when you excavate trees, you tend to find a lot more roots at the surface than you do at greater depth, and particularly a lot more fine roots. It's because that's where the nutrient cycling is occurring. That's where nitrogen is available. They've done studies looking at rooting depth in forest soils, and basically what they find is as deep as the researchers looked, that's as deep as they found roots. And so tree roots do tend to go very deep and can make it through just about anything that you can dig through. So, what I have here is a figure showing water uptake by a root system. And you don't have to worry about the details of the scale or anything on this figure. But with nitrate nitrogen, it tends to readily dissolve in the soil solution because it is an anion and thus isn't tightly absorbed as a cation would be to many of our negatively charged soil surfaces. And so it moves freely with water, and basically wherever trees can uptake water from, they can get dissolved nitrogen to those roots. And so nitrogen can actually move a substantial distance in forest soils before being uptaken by a plant. While I'm saying that, you do have to keep in mind that there's a lot of things competing for nitrogen. Microbes, bacteria, fungi, and other plants. So there is still a competition for available nitrogen, but it's fairly mobile in the soil, particularly in the form of nitrate. Looks like someone has a hand up there. Maybe Sean, you can help with this?
Yeah, it looks like we got a question coming on. I'll
get
your attention as soon as he's posted that if you want to go ahead and continue.
Sounds good. Okay, so that's nitrogen. Nitrogen is fairly mobile in the soil, primarily because the primary form in our southern soils is going to be an anion that moves freely and dissolves readily in the soil solution, which is, of course, primarily water. When we look at phosphorus, here's a figure reproduced from Brady and Wiles' soil textbook, and you can see the pH effects on how much phosphorus is available. Phosphorus is most available at a pH of 6 to 7. This is why they lime agricultural fields, to increase the pH and increase phosphorus availability. Again, that's not a financially viable option in almost any forest soil, particularly in the southern U.S. We tend to have pHs from around 4.5 to maybe 5.5 on average. And so you tend to have very little available phosphorus. We tend to have a lot of altisols that have a lot of clay, and clay has sites where phosphorus can tightly absorb. And once it absorbs to a clay surface, it's much less plant available since it's no longer moving in the soil solution. And so what we end up with phosphorus is similar to what I showed you with nitrogen. And it's shown here in this diagram on the left. There's a lot of phosphorus in a forest soil that is not labile. It does not move. It's tightly absorbed to clays. There's a lesser amount that's labile that can move off those clays or out of mineral form where it may be between layers of mica and become plant available. And then there's far less dissolved in the soil solution. You can see on this figure on the right here how little phosphorus actually moves in a soil. We've got obviously some herbaceous plants here, but what they've done with this autoradiograph is they've managed to look at where the roots of these plants are taking phosphorus up from. These light areas are areas where they have depleted the phosphorus in that soil. You can see they're just growing in plugs right here. These dark areas are areas that still have a high phosphorus concentration. So if you look at this plant that is phosphorus limited, what you'll see is that it's phosphorus limited, but there's a lot of phosphorus left in the soil very close to its roots that it just can't get at. Phosphorus is fairly immobile in soils, again, because it tightly absorbs to clays. And so if we look at how that may play out and how trees have adapted to go get more phosphorus, you really have to start talking about symbioses with mycorrhiza. And so what I have here is a diagram focusing on a plant root here on the left, an area of the soil called the rhizosphere, which is just going to be the area right where all the roots are. If I flip back a slide, you can very clearly see the rhizosphere in this diagram because it's the zone that all the phosphorus has been depleted from. Outside of the rhizosphere is bulk soil. And so very little of the phosphorus in the bulk soil is uptaken. And so over here, we may be able to move some phosphorus from the bulk soil to the rhizosphere, but it's really the rhizosphere where trees are getting their phosphorus from. Mycorrhiza play a huge role in this. And really what they do is they up the rooting density of a tree. If you imagine a 1-inch cube, 1 inch on all sides, the average rooting density of loblolly pine may be about 1 inch of root per a cubic inch of soil. It's a very low rooting density. When you add ectomycorrhizal symbiotes onto that root and all the hyphae, or basically their fungal roots that go out into that soil, you may go from that rooting density of 1 to a rooting density of 970 or so. And so ectomycorrhiza really dramatically increase the rooting density of pines in particular, but many different tree species. And so, imagine a phosphorus in this hypothetical cube way up in the corner. If the pine root is right through the middle of it, it can't get that. As soon as you have the mycorrhizal symbioses, now you may have a hyphae up near it, and it can go and obtain that phosphorus. And so maintaining these symbioses is critical to trees in order to get the phosphorus that they need to grow. There is, of course, a cost to the tree for this. You can see we've got root exudates listed here, including sugars. Basically what happens is that the fungi provide the tree with phosphorus and potentially other nutrients and maybe some water, and in exchange the tree is going to be providing sugars to keep that mycorrhiza alive. And so it is a symbiosis. Trees can get phosphorus in other ways. This says root exudate. All a root exudate is some sort of organic compound, typically, such as citric acid, malic acid, oxalic acid, and others that a root releases into the soil. That initially sounds like it may not be the best idea. I mean, these trees are fixing sugar through photosynthesis. If it's just dumping them into the soil, that doesn't make much sense. They need that to grow. But what we've actually found happen is some of these organic molecules, low molecular weight organic acids in particular, can tightly absorb to the surfaces on clay where phosphorus absorbs. And so they can undergo a ligand exchange reaction whereby this organic acid binds to that clay site and releases the phosphorus that had been bound there. Now, if that happens, that phosphorus becomes plant available and it can be uptaken. So it's a similar trade-off that trees are making with their mycorrhizal symbiotes, in that they're giving up some sugar, some carbon, and in exchange they're getting phosphorus. But if they're most limited by phosphorus and not carbon, that makes good sense from the tree standpoint. Here I've got a couple photos of loblolly pine seedlings, and you can see on the left they have abundant ectomycorrhiza, which you can see down here, this whitish fuzz on the roots. That's a good thing. That means they have high potential to uptake phosphorus. On the right over here, you see they do have normal ectomycorrhiza. There's a little bit of white on there. It's a little reddish here too. You're seeing some pine roots as well there, obviously, but it's obviously not as abundant over here, and these have a lesser potential to potentially uptake phosphorus. And so there's some of the differences in the mycorrhiza that you see there. This is why one main reason why you want mycorrhiza on your seedlings when you plant them. Okay, so we've talked a little bit about how nitrogen and how phosphorus cycle. You could do, a course on just that, so just a very brief overview. Now let's spend a little bit talking about how we assess nitrogen or phosphorus limitation, particularly in a pine plantation. And there's a number of different techniques we'll look at, including chlorosis, foliar concentration, region-specific soil data models, and leaf area index. If we look at chlorosis, what I have here for you is a photo of 2 loblolly pine clones grown in a greenhouse. I should say 1 clone, 2 romettes. And so the 2 trees left and right are genetically identical. They were grown for the same 4-month period in the greenhouse. The only difference is this tree on the left was not fertilized and was grown in a very sandy soil. This tree on the right was fertilized and was grown in the same very sandy soil. You can immediately see the deep rich green color of these needles as compared to the more chlorotic yellowish needles over here. And so a good field forestry technique to assess whether you may be getting some sort of nutrient limitation is to simply look for chlorosis in your needles and see if that may be indicative of nutrient limitation. Obviously, you probably don't want to be making any major financial decisions on whether those needles look a little or more or less green to you. And so let's look at some of these other techniques too. A more robust technique may be to sample some foliage and take it down to your local soil plant and water test lab and basically just have them give you the total needle nitrogen. And if you look here, we've got critical limits. If you're below or at this critical limit, it's indicative that your stand will respond to fertilizer application of that element. If you're above it, it's not as likely to be deficient, and it's less likely that your stand will respond. And so you can see the limiting number there for loblolly pine is somewhere around 1.1 to 1.2% total needle nitrogen content. You really don't have to worry about memorizing the phosphorus numbers because fortunately they're roughly 10% of the nitrogen numbers. So if you know the nitrogen numbers, you pretty much know the phosphorus numbers too. Just move that decimal place over one to the left. That being said, I have seen data sets where stands with a loblolly pine stands with a foliar nitrogen concentration of 1.6% have been fertilized and have shown a 20 or 30% growth response. So this works as a rough estimate. It's not a perfect technique. The next thing we'll look at is a region-specific soil data model. So if we've got anyone here from Florida, this is the CRIF system that you may be familiar with. CRIF is an acronym that stands for Cooperative Research in Forest Fertilization. This is some of Eric Jokola's work out of the University of Florida. I got this slide from Tom Fox. But when you look at a soil survey, there's what, thousands and thousands of soil series in the United States. What we really care about as foresters is how do we manage that soil to meet whatever our forest management objectives are. And what the CRIF system does is it distills all these series down into groups, basically A through H, I believe, and gives you some simplistic management recommendations for each of those groups. And so if we look at an example, here is CRIF Group A. And CRIF Group A, I'm not going to get into too much detail with this system, but if you categorize your soil as a CRIF Group A, it's likely to be severely phosphorus deficient and can respond dramatically. You may double growth response over the next rotation with a single establishment application of phosphorus. These soils tend to be on old Pleistocene terraces, soils derived from that geological material, and they tend to be in a band near the lower coastal plain. And so that's an example of a region-specific soil data model. Of course, many large forest corporations and forest industry, they've put together models for specific areas where they have a lot of land, and some of these involve things such as depth of the water table, soil texture, a number of different variables that do a pretty good job within the region that they're parameterized for of predicting what nutrients may be limiting to a site. The other thing that large forest products corporations are doing today that seems to be working pretty well is using aerial imagery. And so here I have a Landsat image of East Texas taken from the literature for you. You can see, of course, it's a Landsat image. So these red stands are our pine stands. And what they found basically in this work they did published in 2006 out of NC State primarily, is that there was a very good relationship between some data you can extract from a Landsat image and on the left here, leaf area index. So we're already seeing that good relationship between leaf area index and stand growth, and now what we're seeing is a good relationship between the simple ratio you derive from Landsat image and leaf area index, and what that's going to allow you to do is this. You can categorize your stands if you're working for a large TMO or a REIT, say, and you can figure out here we've got stands with lower leaf area index in white. Here we've got stands with intermediate leaf area index, 2.3 to 3.3 in the brighter red, and then in this more maroon sort of red, we've got our high leaf area index stands. And so you can see very, very quickly what they can now do is if they know, they want to fertilize, say, 30,000 acres this year, they can look at their 500,000 acre tree farm or whatever it might be and prioritize. We want to fertilize this stand, this stand, this stand, and this stand. These stands are last priority for fertilization because we don't think they're going to respond to fertilizer additions. And so this is a very quick way to get a broad understanding of leaf area index that can then be used as a surrogate for nutrient limitation. You still don't know what nutrient is limiting these stands, so you would still need some of that information from either foliar vegetation analysis or more likely from some other sort of soil workup. Okay, so a review of objective 2 is you have to keep in mind that nutrient cycling differs depending on what nutrient you're talking about. It varies depending on the soil texture. Phosphorus, is going to be able to move further in a soil with less clay than it is in a soil with more clay. Most of the nutrients in a soil are not available to the trees. If you're doing a soil analysis that just gives you total nitrogen or total phosphorus, that may tell you very little about how much is actually becoming plant available. And then when we go to assess nutrient limitations, again, you have to understand how these nutrients cycle and what that assessment technique is actually telling you. So that completes the second of 3 objectives here. Anyone have any questions on objective 2? I see we're nearing 4 o'clock here. Sean, should we proceed, or what would you suggest?
Yeah, go ahead and proceed. In the interest of time, when we hit 4/5 o'clock, I'll go ahead and push out the webinar. So just go ahead.
Okay. Okay, we've got a couple more pop quizzes here, and so what we're looking at now is climate change. How's that going to impact nutrient cycling? And I'm focusing solely here on elevated atmospheric concentrations of carbon dioxide. And so compared to now, climate change is projected to have what effect on forest nutrition in the short term? And by short term, I mean a period of, say, 5 to 10 years. So just a few years, less than 1 rotation up to maybe 1 rotation. Is elevated atmospheric CO2 going to increase nutrient availability, decrease nutrient availability, cause no change in nutrient availability, or we really don't know yet, D? So take a minute and let's see what folks think. Okay, so it looks like the consensus is, with those that responded, is we really don't know much yet, with second being increasing nutrient availability. So let's look at some of the data on this. Before we do that, let's ask one more question. Same question now, let's think longer term. Say 100 years or several rotations. So in the longer term, what's going to happen? Take a minute and answer this one. Okay, looks like most folks think we don't know much yet, and then by far the second one is actual decreases in nutrient availability. So let's go look at the data that we have so far and see what the scientific consensus seems to be thus far. What I've got here is one of these hockey stick graphs. What we're looking at is year 0 to year 2000, and we're looking, I'm not focused as much on methane or nitrous oxide, just this red line, carbon dioxide. And so prior to the Industrial Revolution, atmospheric carbon dioxide concentrations were about 275 parts per million. This data only goes through 2000, so it's up here at 380. We're over 400 parts per million now. Whatever your personal beliefs on climate change are, this is pretty ironclad scientific evidence. We know that atmospheric carbon dioxide concentration has increased. There's pretty much no debate on that. And so this is what I'm going to focus on for this portion of the talk, because we have more data on this. Here's a paper that came out in 2004 hypothesizing that with elevated atmospheric carbon dioxide, we would develop what they called a progressive nitrogen limitation of ecosystems. And how their theory worked is when you increase CO2, photosynthesis works more efficiently. And so that increases net primary productivity, which is just growing biomass. We grow more biomass with higher atmospheric CO2, and that seems to hold up. There's little controversy there. What their theory then stated is we're going to input more carbon into the soil, both in the terms of maybe root exudates, but also just we're going to grow more roots because we're growing more biomass. And what's going to happen when you put more carbon into the soil is you're going to sequester more nitrogen. As you sequester more nitrogen, there's going to be less available or labile nitrogen. Nitrogen availability goes down, nitrogen uptake goes down. You see the negative sign down here, and nitrogen becomes more and more limiting because you've got it all sequestered in either soil organic matter, SOM, or biomass in leaf litter. And so the theory here was, as atmospheric CO2 rose, we would see more and more of these nitrogen limitations. So that argued, I believe it was with choice B there, short-term decreases in nitrogen availability. Here's another figure from that paper when they looked at the actual data. And this is sort of a complicated graph because they keep switching these lines back and forth, but we'll go through and interpret it. And they basically put up a two-stage sort of question to determine if P&L, or that progressive nitrogen limitation, was occurring. And so their first question was, if atmospheric CO2 goes up, do we actually grow more biomass? Is NPP stimulated? And basically, if NPP is not stimulated, if you don't get more growth, I'm following this line here to see no, nitrogen limitations don't develop. Nothing's really changed from the perspective of soil cycling, and we found examples where that's true. And there are examples such as the Nevada desert, where they're not carbon dioxide limited, they're water limited, or the Alaska tundra. That's obviously going to be a system that's temperature limited and limited by the length of the growing season. If growth is increased, we follow this line here, the next question is, do we have an increase in nitrogen demand? And we believe that we do because you're growing more. And so they're not asking a question there, but the next question is, can the nitrogen supply from the soil meet that increase in demand? We've seen some examples where it can, where it cannot. If we follow this, this is the no line here. In Texas grasslands and Florida grasslands, they basically found that, yes, those stands do become more nitrogen limited. So that's an example where nitrogen limitations have occurred in the literature. In some cases, such as the Kansas prairie, nitrogen supply does meet the nitrogen demand, and you don't become nitrogen limited. Remember back to that mollisol data we looked at, how they had a lot of nitrogen in those mollisols? This makes sense because these are soils that have a lot of nitrogen. And so those soils did not limit those ecosystems from a nitrogen standpoint yet. And so what we're seeing, the only examples of forest are really right over here, and they are observing nitrogen limitations occurring. So what I want to look at now is some more in-depth than just those one or two studies, examples of where we've tried to assess the impacts of elevated atmospheric CO2 on nutrient cycling.
Excuse me for a second, Jeremy, just if I can interject. I'm going to go ahead and push out our post-webinar survey just in case anyone is pressed for time. So just one moment. And this should open up a window in your browser. Give it just a second. It might be just a little slow. All right, go ahead.
Okay, so what you're seeing here are photos of the Duke FACE site. And FACE is an acronym for Free Air Carbon Dioxide Enrichment. And what they did, you see these rings of towers? They put these rings of towers in, and what these rings of towers did is emit carbon dioxide. And what they did is they increased the ambient atmospheric concentration by 200 parts per million. And they didn't do this in the short term. They did this for a period of 15 years in a loblolly pine plantation. And so what we have is a relatively long-term data set from a pine plantation where they've created atmospheric carbon dioxide concentrations we don't expect to see for centuries from now. They have other treatments too where they took half of each of these rings and they fertilized one half and didn't fertilize the other half. And so they carried this on for a long time, and we have really an enormous amount of data from these trials on just exactly how nutrient cycling is going to respond to increased atmospheric CO2. And here's what they found. So on this graph here, first off, see these open circles down here? Okay, those are the ambient CO2 plots. The closed circles are elevated CO2. And so on the y-axis, what we have is NPP. That's growth. Okay, you could change these units and put it in there, in terms of, square feet per hectare, basal area, or cubic feet per acre. So that's basically just volume we're seeing on the y-axis. And what we see is with elevated CO2, sure enough, you get more growth. We expected that. What I want you to see on the x-axis here is this nitrogen availability index, the amount of nitrogen available in grams per square meter of land. And what you'll notice is, in general, these black dots have shifted to the right, and the line they're forming is steeper. And so what this data is showing us is that there's greater available nitrogen under a scenario of elevated CO2, even considering for the fact that these stands are growing more biomass. This data right here does not support that progressive nitrogen limitation theory we just discussed. It appears that there's plenty of available nitrogen from the period 1997 to 2004. What I'm showing you here is another sort of complicated figure. See these triangles over here? That's our Duke face site again, but we've combined it with 3 other free air enrichment, carbon dioxide enrichment trials. These diamonds here, that's Oak Ridge National Lab. Those are sweet gum stands. These squares right here, that's Pop Euroface. Those are different species of poplars. And then this Rhinelander down here is a mixed hardwood forest in Germany. And basically what you see on this graph, again, we have nitrogen availability right here in terms of uptake and net primary productivity, so again, growth on the y-axis. And what you generally see is the open triangles down here, the closed ones up here, along the same line, which represents a nitrogen use efficiency, or basically how much nitrogen does it take to grow a given amount of biomass. And so what you see is they shift up the same line for the triangles, which means you're getting more growth, and nitrogen's not becoming limited. You have the same nitrogen use efficiency. Same thing for the diamonds, which are going to be sweet gum in Tennessee. Same thing for the circles, which is mixed hardwood trees in Germany. Over here, what they found for Euroface, the poplars in Europe, is that they shifted straight up. Nitrogen use efficiency actually increased for those, where it didn't for the others, but it turns out that's not a nitrogen-limited system, so it's not as analogous to our southern pines. And so again, all across the world, all across the southern United States, different species, we're seeing pretty much the same thing from these space trials. And again, if you think about why, it makes a lot of sense. If you're thinking about phosphorus again, and phosphorus availability, where they found similar trends to what we just discussed for nitrogen, basically the trees have plenty of carbon. They're getting all sorts of extra carbon from the atmosphere with elevated CO2, and so what do they do with it? They pump it out in the soil. And they basically hand it off to mycorrhiza, which help them get more phosphorus, or they pump it out in the form of root exudates, which undergo those ligand exchange reactions, which ultimately get them more phosphorus. There's something called a rhizosphere priming effect. That's how they think they're getting more available nitrogen. Basically what's going on is they dump these sugars out of the roots because they have more carbon than they need. What those sugars do is they stimulate the microbial community. And what the microbial community then does is decompose organic matter at a more rapid rate, which mineralizes nitrogen, which makes it plant available at a greater rate. And so really, trees are using all this extra carbon dioxide not just to grow bigger, which they are, but also to dump into the soil to prevent any nutrient limitations. And so what we're seeing from these space sites is, at least in the short term, nutrient deficiencies don't appear to be developing. Here's one more graph from that Duke face site that shows you again what I just indicated. Let's look down here. This is basically root exudation. Okay, under the fertilized plots where nitrogen was highly available, root exudation is low. It's higher over here where they didn't fertilize and nitrogen was limiting. And so the trees sort of know, if you want to think of it that way, when nitrogen is limiting, and they exude more root exudates in response. The other thing you'll notice is under elevated atmospheric CO2, there's no significant difference there, but under low nitrogen there is. They actually exude more carbon out of the roots in terms of root exudates when they have more from elevated CO2. And so if you look at that, in the short term at least, it appears that trees will be able to modify the soil environment by how they handle root exudation and symbiosis with mycorrhiza to get all the nutrients they need to fuel greater growth rates. Now in the longer term, we know a lot less. I would argue that choice D is probably the best answer right now. You certainly have to ask the question, well, if they do all this in the short term and they free up all these nutrients that are sequestered in organic matter and in the soil, won't that become limiting in the long term? It may well. We really don't know yet. The other thing I would argue you have to keep in mind everything we just looked at is focused solely really on elevated atmospheric CO2. We're not talking about any of the other potential impacts of climate change, such as changes in temperature or increasing temperatures in some places, and changes in precipitation. So those could also play a major role. For example, in the West Gulf here, if 100 years from now, we have higher evapotranspiration rates because it's a little warmer, we're getting a little less rain and more prolonged droughts, well, that obviously may make water much more of a limitation here than it has been to date. So you've got to keep some of those other features of climate change in mind and how those might interact with elevated CO2. Okay, so that was a quick third section, but basically, in the short term, trees are able to increase rates of nitrogen and phosphorus cycling due to that carbon fertilization effect. You're essentially fertilizing the trees with carbon, even though we don't typically think of that as a nutrient. That's going to be confounded with temperature and water in the future, and so our longer-term impacts are still largely unknown. Globally right now they're installing a second phase of phase trials where they not only increase atmospheric CO2, but they actually have heaters in the soil, and so they're warming up soil temperatures just to see what happens there. And so we may understand more of those interactions in the future. The long-term impacts are going to be trickier to wrap our heads around, I would argue. So that's basically the three main topics I wanted to hit on, and so just a very quick review. Nitrogen and phosphorus appear to be the most limiting resources to growth in our southern pine forests. Nutrients cycle differently, and you really have to understand how each nutrient cycles in order to understand how deficiencies may develop and how you can ameliorate those deficiencies. And in the short term, it appears that some features of climate change may not have a negative impact on forest nutrient cycling, but again, those longer term impacts are unknown. And that's all I've got for you, so I'd be happy to field any questions anyone who's left may still have.
While we're waiting for questions, let's give them a round of applause. If you click on or hover your mouse over the smiley face under your name in the participant sidebar, you can select the applause icon. And if you have any questions, please type them in the chat box below.
Seem to have a hand up again from Dwight Batts. We can't hear you if you talk. It doesn't have the microphones enabled on everyone.